Hello everyone, welcome back to the channel. If you're new here, my name's Zach Hancock. I'm an evolutionary biologist who specializes in population genetics, phylogenetics, and genome evolution. And today, we're going to be talking about one of the most persistent creationist claims, the waiting time problem. We're going to take a broad historical view, looking at the very first waiting time papers, some of the controversy that it caused, and then we're going to talk about some of the ones that have appeared in more recent years. We're going to show how the waiting time problem actually is not a problem for evolution at all, and that it relies on a lot of unrealistic assumptions and preposterous simulation and mathematical models. So buckle up and let's talk about the issues and the history and controversy behind the so-called waiting time problem. The saga of the waiting time problem began in 2004, 8 years after the biochemist Michael Behe published Darwin's Black Box, in which he put forward an argument from incredulity that he dubbed intelligent design. A paper was submitted to the journal Protein Science in early April and arrived on the desk of the journal's editor, Mark Hermansen. The paper was penned by Behe and a physicist named David Snoke. The opening line of the paper read, quote, "Although many scientists assume that the Darwinian processes account for the evolution of complex biochemical systems, we are skeptical." Hermansen and the other editors at Protein Science were well aware that Behe was associated with the up-and-coming movement of intelligent design spearheaded by the Discovery Institute. Hermansen, in an editorial on the history of the paper's submission and the backlash that followed, remarked that the editors felt that they were in an uncomfortable position regarding how to handle the piece. On the one hand, they felt strongly that intelligent design was not science, but on the other, they felt that rejecting Behe out of hand would be perceived as unfairness. Thus, they made the difficult decision of sending the paper out for review. The paper was in review for 2 months, after which it was accepted for publication. Shortly after publication, letters began to pour into Protein Science, both from regular readers and from biologists in other fields, to voice vehement disagreement with the article. Multiple papers have been published in the years since addressing it, but the first formal response was by population geneticist Michael Lynch. Lynch reached out to the journal after seeing the article and asked if they'd be interested in publishing a rebuttal. The editors agreed, and what followed was a scathing refutation by Lynch in September of 2005. In the same edition, Behe and Snoke published a response defending the 2004 paper. Hermansen opted to publish a short editorial on the fiasco in that same volume and remarked how the editors had toyed with the idea of letting readers respond in short letters to the journal, but ultimately decided, quote, that no useful purpose would be served. Almost certainly, they realized that the conversation would dissolve into polemics, and so it ended there, at least as far as Protein Science was concerned. Hermansen ended his editorial by remarking that Lynch's approach to addressing Behe and Snoke was the, quote, desired scientific approach, and ends by quoting Bruce Alberts, who writes, "In evolution, as in all areas of science, our knowledge is incomplete. But the entire success of the scientific enterprise has depended on an insistence that these gaps be filled by natural explanations, logically derived from confirmable evidence." Of course, that would not be the end of it, and the paper would return to the spotlight less than a year later in the famous Kitzmiller v. Dover trial in Pennsylvania, in which intelligent design would quite literally stand trial. So what was the central content of Behe and Snoke's 2004 paper? Why was everyone so upset? Is there something inherently wrong with challenging established paradigms? And how has their ideas fared in the 20 years since they first proposed them? The crux of their 2004 paper, titled Simulating Evolution by Gene Duplication of Protein Features That Require Multiple Amino Acid Residues, I know, a mouthful of a title, was that the origination of a novel protein interaction that required multiple specific mutations would take an inordinately long amount of time. We're talking billions of years. They called this the waiting time problem. Let's try and understand this idea at a simple level. Pictured here is a string of nucleotides that code for some hypothetical protein in a population of microbes. Let's say this population is suddenly subjected to antibiotics and it needs to rapidly respond. Further, the particular nucleotide bases that need to change are those marked. These very specific changes are required for this microbe to persist. How likely are these two changes to occur? There are many factors we need to consider. First, we need to know the rate at which mutations happen at all. Second, we need to know the impact of each of these mutations on their own. If you only get one of these mutations, are you at an advantage, even if it's smaller than if you have both? Or are both required to be advantageous at all? Or even more restrictively, if you only have one of them, maybe you're worse off than if you have both, and thus both must occur simultaneously to be advantageous. Finally, we need to know something about the population size, which tells us what the expected pool of standing variation might be, and further, what's the rate of genetic drift. Let's start with how long we have to wait for a specific mutation to occur. If we assume that the rate of mutation is 1e to the negative 8 and a genome size of 1e to the 9, or a billion, then the probability that a specific mutation occurs is 1 over 1e to the 9 multiplied by 1e to the negative 8. Now, there are some individuals in the population, denoted in which this can occur. So we multiply this by the population size. Let's call it 1,000,000. Thus, the probability per generation of getting a specific mutation is 1e to the negative 11, given the parameters that we've specified. That means that we expect it to take approximately 1e to 11 generations to get one very specific mutation. In humans, that amounts to 2.5e to the 12 years, as in longer than the universe has been in existence. It's pretty easy to see how this can be taken to mean that evolution couldn't happen. The retired Cornell geneticist and author of the book Genetic Entropy, John Sanford, has argued that since humans and chimpanzees have millions of differences between us, evolution via mutation plus selection, what he calls the neo-Darwinian theory, is impossible. There's simply not enough time in recorded history. Behe and Snoke's model is a bit more complicated than this, and I want to take a moment to explain how it works. They imagine a haploid population in which individuals have a duplicate for a protein, and that either of these, the parent or the daughter copy duplicate, can undergo either the necessary mutations to achieve neo-functionalization, that is where the duplicate acquires a novel function that's necessary for the microbe to persist, or they can undergo a mutation that causes them to degrade and what they call become a null allele. That is, where the duplicate loses function altogether. They imagine that in these duplicates, only specific sites can generate a novel protein, but any site can lead to the duplicate losing function. In their model, individuals can have any number of duplicates, or they can have no duplicates. But importantly, the duplicates are initially identical. That is, though everyone in the population has potentially one or more, none are permitted to mutate prior to reaching this high frequency. In this way, there's a sort of mutational race between neo-functionalization and the emergence of a null allele that would cause one of the duplicates to become non-functional. You can probably already guess their results. They found that to fix 3 specific nucleotides in 100 million generations would require a population size of 100 billion individuals. To do the same thing in only 1 million generations would require a population size of 10 raised to the 17. So evolution dead, right? Clearly the waiting time problem is a serious impediment to Darwinian evolution. Should we hang up our hats and start considering divine intervention as a legitimate hypothesis? If you've been paying attention, you might have picked up on a few blatant problems already. Let's start with the most fundamental and then work our way to the specific. All the waiting time papers, of which we're going to discuss a couple more, rely on what I call hindsight evolutionary thinking. That is, they begin with an observation in the present and ask, how likely is that event to have occurred? This might seem like a logical line of inquiry on the face of it, but a simple demonstration shows why it's faulty. Imagine I present you with 100 numbers ranging from 1 through 4. Given that string of numbers, you can ask, how likely is it that I got those numbers? Well, if you assume that I picked them randomly, and then you calculate the probability of that string as 1 over 4 raised to the 100, that equals 6.22 e to the negative 61. That's a number so small as to be virtually indistinguishable from 0. So what would you conclude? You might say, well, I must not have picked them randomly at all. I must have picked them deliberately. An intelligent design choice, if you will. And you'd be wrong. These numbers were picked using a random number generator. All I specified was to pick numbers 1 through 4 and to draw it 100 times. Notice that any string of numbers that were picked are equally improbable, but some number will be picked. This is key to understanding evolution, and importantly, its reliance on historical contingency. Each time I run the program, I'll get different numbers. This is reminiscent of paleontologist Stephen Jay Gould's point that if we rewound the tape of life and started over, we'd get dramatically different results. Evolution has no goal. It's not trying to get a specific thing. What it produces is contingent on the standing variation it has to work with. If that variation at its disposal is insufficient to survive in the given environment, the population goes extinct. Indeed, extinction has been the fate of the vast majority of organisms that have ever lived on the planet. Thus, from the outset, the question is malformed. Further, even if we assume that evolutionary processes were trying to get us somewhere, we should recognize that one of the principal findings of proteomics is that there are many ways to produce the exact same result. That is, there's no single road to Rome. There are many, some easier to traverse than others. Evolution is the drunkard who doesn't know where he's going or that he's trying to get to Rome at all. He's wandering the paths, and if some paths are smoother and softer on his boots, then those will be selected until, lo and behold, he looks up and realizes he's in Rome. Let's get into some specifics now. The major rebuttal of Behe and Snoke comes from Michael Lynch, and he begins by pointing out the major flaws in their model. First, he notes that they assume that the duplications emerge and are fixed prior to any diversity being permitted to accrue in them, as I stated previously. That is, as the duplication is going to fixation, they prevent any mutations from occurring on them along the way to fixation. By separating these two events, fixation and origination, they unrealistically exacerbate the time to novelty. Second, Lynch criticizes their assumption that most mutations in the duplicate gene would cause loss of function. It has been known since at least the 1970s that protein-coding genes are quite tolerant of amino acid substitutions. He cites one study that found that 44% of the codons in the lac repressor gene of E. coli tolerate substitutions. Another study found that a mere 14% of amino acids in a bacterial ribonuclease enzyme can cause inactivation when mutated. In humans, the DNA glycosylase enzyme can withstand 66% of its amino acid sequence being replaced and still retain function. Third, Lynch notes that Behe and Snoke assume only 2 specific codons are capable of generating the novel function. He states that by allowing even 10 codons out of an average of 450 in a typical protein would increase the probability of neofunctionalization by 10-fold. Again, this is the many roads to Rome analogy from earlier. Lynch then constructs a model that he contends is far more biologically realistic to further challenge Behe and Snoke's conclusions. In his model, the duplications are not fixed, but instead emerge and go to fixation. In this way, along the way to fixation, duplicates can acquire mutations simultaneously while increasing in frequency. Furthermore, citing studies already mentioned that most proteins can sustain a wide range of amino acid substitutions without losing any function, he permits many substitutions in the duplicates to be neutral in the intermediate steps along their way to neofunctionalization, instead of assuming that all of them would result in a null allele. The results of Lynch's model are in stark contrast with those of Behe and Snoke. He found that the probability of neofunctionalization was actually very high in populations of around 1 million individuals and could occur in approximately 1 million years, as opposed to 1 billion that was required by the model of Behe and Snoke. Furthermore, Lynch notes that Behe and Snoke appear blind to the fact that duplicates actually provide a weak mutational advantage that's equal to the rate of mutation to null alleles. While drift dominates if n is less than 1 over u, where u is the mutation rate to null alleles, if 2nu is greater than 1, then the mutational advantage alone can maintain duplicate alleles. This is because for individuals with 1 or more duplicates, they are actually buffered against mutations that would cause one of the alleles to lose function, whereas those without a duplicate will just die if they get a mutation to a null allele. Again, while this effect is likely very weak in organisms with population sizes less than about 100 million, for microbes, which is who Behe and Snoke are specifically modeling because they're using haploids, 2nu is almost always greater than 1. And hence selective pressures alone can cause the persistence of duplicates, dramatically increasing the probability of neofunctionalization. For organisms with smaller population sizes, Lynch points out that subfunctionalization is actually more likely, again something that's not allowed in Behe and Snoke's model. But even this saves the duplicates. Once loss of function alleles appear in each one of the duplicates, though not at the exact same site, each of the allele is then buffered against additional mutations because it would result in a total loss of function. Again, this solidifies the duplicates, ensuring they will persist and allowing ample time for neofunctionalization to occur, unlike in Behe and Snoke's model that assume that neofunctionalization must occur before they become non-functional, with no potential for subfunctionalization. Several recent studies have actually advocated a subfunctional to neofunctional model for the origin of new genes. That is, after duplication, genes rapidly will subfunctionalize, but then gradually will acquire novel functions thereafter. Hence, neofunctionalization proceeds from subfunctionalization. Lynch pulls no punches, writing, quote, "Given that the authors restricted their attention to one of the most difficult pathways to an adaptive product imaginable, it comes as no surprise that their efforts did not bear much fruit." And, quote, "that the contrarian interpretations of Behe and Snoke are entirely an artifact of incorrect biological assumptions and unjustified mathematical oversimplification." To reiterate, Behe and Snoke introduced the waiting time problem, using hindsight evolutionary thinking that all but guarantees exceptionally low probabilities of literally anything at all happening. They employed a mathematical model that makes bogus assumptions about biology, one so restrictive that it all but guaranteed at the outset to produce the results that Behe and Snoke wanted to see. September of 2005, in which Lynch's rebuttal and Hermitson's editorial would appear in Protein Science, happened to be the same month that the opening arguments began in the Kitzmiller v. Dover trial, in which Behe served as an expert witness on the side of intelligent design. During the trial, Behe cited his paper with Snoke as evidence that biological complexity was unlikely to evolve. However, under cross-examination, he was forced to admit that, quote, "the number of prokaryotes in 1 ton of soil are 7 orders of magnitude higher than the population size that it would take to produce a disulfide bond," under his and Snoke's model, and that while bacteria may not evolve the disulfide bond in 2 years in a lab, 3.5 billion years would be more than enough. Thus, in Behe's own stated testimony, the waiting time problem is not, in fact, a problem for evolution. Several other papers were published over the years, debunking the Behe and Snoke paper, including one by geneticist Joanna Masel, the mathematician Rick Durrett, and an additional paper by Lynch in 2010. Despite the clear crash and burn of this idea, the Discovery Institute continued to cite it as evidence for intelligent design. While the Institute became increasingly irrelevant after their stinging defeat in Dover in 2005, a new champion of the idea emerged in 2015. This champion is the aforementioned retired Cornell professor, John Sanford. His group had designed and released a forward time simulator in 2007 called Mendel's Accountant. The program has been hailed among creationists as the most advanced simulation software in existence. Of course, this is not merely hyperbole, but borderline a downright lie. The program EasyPop had already been around for 6 years by that point, and Mendel brought little more to the table. And by 2005, SimuPop had been published. And each of these programs during the time of their heyday experienced much greater use than Mendel. In fact, only Sanford's group and other creationists actually used Mendel for scientific publications. Be that as it may, Sanford turned his overhyped program towards the waiting time problem in 2015. With a few tweaks, they were able to set up a simulation in which a string of letters of a given size were permitted to mutate. They pre-specified a target sequence that represents the novel protein and then waited to see how long it would take for mutations to generate the sequence. As you might expect, just like Behe and Snoke, they found waiting times in the billions of years. But unlike their predecessors, their paper did not receive any letters of detestation or backlash. In fact, if you ask most people in the field, they wouldn't even be aware that a second waiting time paper had ever been published. So what gives? Did the evolutionary community see the error in their ways, that the Sanford paper was just so much better that it just shut them down once and for all and the only option was to ignore it? No. The paper is just really, really bad. So bad that to dignify it with a response would give it wider readership than it would have ever had otherwise. You see, Behe and Snoke published their paper in Protein Science, a reputable journal with a wide readership. The Sanford paper appeared in the Journal of Theoretical Biology and Medical Modeling. The journal only had 18 volumes since it was established in 2004, and it went under in December of 2021. Prior to a creationist citing this paper, I'd literally never even heard of the journal. If you're not super familiar with scientific publishing, you should know that there exists hundreds of journals in every single discipline. Literally anyone can start a scientific journal and even get big publishers like Springer to pick them up. Journal publishers don't care about science, they're in it for the money. Editors of journals guide the direction and the scope of the journal and dictate the rigor of the peer review and publication process. Most journals are bad. They publish shoddy work with little to no peer review and charge exorbitant publishing fees. In the scientific community, we call these predatory journals. That is, they're low-quality journals that will publish literally anything you send them so long as you pay the publication fee. Now, that's not to say that there's no peer review at all, but rarely, if ever, do people actually qualified to peer review the work agree to do so. Most scientists refuse on principle to peer review for predatory journals. So often the editor ends up doing the review, or someone reviews it that's just totally outside of the field in question. All of this is well known in the scientific community. We have dozens of journals in each field that are respected due to their high standards, intense peer review process, and the quality of studies that they publish. For someone not familiar with any of this, though, it can be hard to know what a good journal is versus a predatory one. These predatory journals often masquerade as legit journals, so how can you possibly tell the difference? Here's a tip. When in doubt, see if the journal is published by a society. Scientific societies include the American Naturalist Society, ASN, the Society for the Study of Evolution, SSE, the Society for Molecular Biology and Evolution, SMBE, and the Genetic Society of America, GSA, etc., etc. Each scientific society has one or more journals that they sponsor and publish, and they tend to be held to the highest degree of rigor. With all that said, let's not just hand-wave away Sanford's paper solely because it was published in a shoddy journal. Let's actually evaluate the scientific merit. Unlike the Behe and Snoke's paper, there has been no scientific response to the Sanford one that we can lean on, hence we're going to have to play the role of reviewer on this one. Sanford and friends waste no time blowing the creationist dog whistle. The very first line claims, quote, "At the heart of all information systems, including biological information systems, are strings of symbolic characters." They go on to argue that DNA is like strings of letters, and these strings of letters, under a Darwinian paradigm, must emerge via the so-called mutation selection process. That is, a random mutation must first produce the functional variant that selection then promotes. They also spend a paragraph just absolutely butchering Haldane's cost of selection and Kimura's motivation for the neutral theory, two things that I've already spent considerable time talking about on this channel. See my video response to Chris Roop and Donnie Badinsky's article on genetic entropy if you want to know more about that. Now, the Behe and Snoke and subsequent papers on the waiting time problem focused on microbes with exceptionally large populations, but instead, Sanford and pals are concerned with human evolution. They write, quote, "In this paper, we address the question, how long does it take for the simplest biological text strings to arise and be fixed within a hominin population?" They go on to set up the problem by stating that humans and chimps are at least 5% diverged genetically, corresponding to around 150 million base pair differences. Thus, they write, quote, this substantial genomic difference implies many new substrings within the human genome." You can already see where this is going. These new text strings must have been purposeful. That is, they must be contributing to our differences, and since they are here in front of us, we can ask, how likely is it that these specific changes occurred? You should recognize that phrase from our simple example previously on the flaws of hindsight evolutionary thinking. Sanford and buddies assume from the outset that the changes that occurred must have happened. They seem incapable of grasping that there's no reason these specific changes were the ones that emerged. Again, as Gould stated, rerun the tape of life and we get a totally different set of changes. Their model is much simpler than Behe and Snoke's. They imagine a string of between 2 and 8 nucleotides that they designate A's. They have some target, let's say T's. They specify a given mutation rate, ranging from 0.01 to 1e to the negative 5, which is actually much higher than the biological rate of around 1e to the negative 8. They do this, they state, because simulating the real rate would be too computationally costly and would require them to simulate billions of generations. So they just multiply their results by a correction factor, which is effectively an extrapolation to what the result would be at a more realistic rate. They write concerning Mendel's accountant that it, quote, "faithfully models our present understanding of the neo-Darwinian mutation selection process and maintains biological realism at the level that has not been previously possible." Again, as I've already mentioned, other programs existed prior to Mendel with the exact same capabilities, but beyond this, I'm extremely skeptical of their claim that it faithfully models anything. For example, I ran Mendel through a simple sanity check. In population genetic theory, the expected amount of genetic diversity in a population, given no selection at all, should be equal to the mutation rate multiplied by the population size 4n mu. So I set up a simple neutral simulation in Mendel to see what would happen. I found that it equilibrated at a diversity almost 10 times higher than it should have. I'm unsure if this is a bug or if it's designed to be this way, but that immediately raised some red flags. Next is that Mendel has a default parameter called heritability. In quantitative genetics, heritability describes the proportion of the trait variance that is due to genetic variance as opposed to environmental variance. Mendel sets this value to a default of 10%, and it treats it as a kind of scaling factor on any beneficial mutation. For example, if your input of a selective benefit of a mutation is 20%, that's not actually the benefit default setting will give you. Instead, it's downscaled by the heritability parameter, weakening selection considerably. This is likely why all default models in Mendel show fitness declines. A constant low heritability makes positive selection so weak that it can't keep up with the deleterious mutation accumulation. This presentation of heritability is at best wrong and at worst designed to be misleading. Heritability is not a fixed quantity, and it is not prescriptive, it is descriptive. For example, imagine an organism that enters a new environment that possesses a trait in which most of the variance in that trait is determined by genetics, i.e., it has a high heritability. But in the new environment, that trait is strongly selected upon. Over time, selection removes genetic variation as it molds the trait to fit the new environment, and so the genetic variance, and hence the heritability, both decrease. Eventually, nearly all the variance in that trait will be explained by the environment, and that trait will have a very low heritability. Thus, selection itself reduces heritability, and so to treat heritability as a fixed quantity that actually dictates how selection acts is grossly misleading. Lastly, the only distribution of mutational effects that Mendel offers is the Weibull distribution. There are many probability distributions. You're likely most familiar with the normal distribution, which is defined by the fact that 98% of all values fall within 3 standard deviations of the mean, generating this kind of bell curve shape. Other distributions take on other shapes, and these shapes are determined by what is called the shape parameters. The Weibull is no exception, but in Mendel, you can't designate what the specific shape parameters are. This means that there's some designation under the hood already, but they aren't made explicit to the user. Hence, it's impossible to know what the default distribution of mutational effects looks like in Mendel. Again, this feels like having the veil pulled over your eyes. As users, we should have full control of the distribution, and without this knowledge, it's impossible to judge the outcomes of our models. Outside of these weird quirks of Mendel, a major issue emerges from the paper itself, seems to be due to Sanford's confusion about what constitutes genetic drift versus selection. Several statements from the paper make this clear. He writes, quote, "Fertility rate was set at the default setting such that 50% of all progeny were selectively eliminated every generation, reflecting very intense selection." But this is not selection, because it's not connected to fitness. As they describe in their methods, the 50% that are chosen are picked at random. Hence, this reflects simply genetic drift, not selection. Selection plays no role in their simulations until one of the mutations arises on the string that matches the target. Then, their model grants that individual a fitness advantage. But how that fitness advantage is realized is bizarre. In most forward time simulators, selection is either soft, acting on reproduction, or hard, acting on survival. Soft selection simulations are when the population size is kept constant, so fitness represents the probability you have offspring. Hard selection occurs in non-Wright Fisher models, in which the population size is not expected to stay constant, and fitness is the probability that you survive to reproduce. Mendel does something totally different. They write, quote, "In the selection subroutine, each individual that has a non-zero value in the target match array receives a fitness bonus specified as a user-specified parameter. During the selection phase, the individuals are sorted according to fitness, and the top NP individuals are selected to reproduce the next generation, where NP is the user-specified population size. NP is assumed constant in cases we consider." From my reading of this, it seems like they sort individuals every generation randomly, except for those that have sites that match the target sequence. Those are then placed on top of the list. Mendel then purges the bottom 50% to maintain population size. But this can't be correct, because it would imply that any fitness advantage would place individuals at the top of the list, ensuring that they always reproduce. This would negate their point in the results that many new mutations are lost by chance. So something else is going on, and that's the thing. I can never figure out exactly how Mendel is running. The methods are so poorly written, with the model described in ways that are so non-standard to like how models are normally described in the scientific literature, that I just struggle to follow the order of events. Regardless, their results clearly indicate something is fishy. They simulate fitness increases over 4 orders of magnitude, but the rate of fixation barely changes by 3, with even a fitness advantage of 10% taking 8.4 million years to fix on average once it emerges. Another bonkers thing that makes me think something weird is going on in Mendel is that they're finding that in simulations with low mutation rates, they observe what they called drift-induced string homogenization. That is, that little variation was being maintained in the population due to drift, extending the waiting time problem for the low mutation rate situations. Indeed, they write, quote, drift was eliminating variants about as fast as new mutations were happening. This was resulting in near zero genetic variance of the string within the population. Let me explain why this should have been an obvious red flag. First, if drift was eliminating variants as fast as new mutations were happening, the population would not be at near zero genetic variance. This is actually what's called mutation drift equilibrium, and given the settings that they enforced, they should have observed 4NU genetic variance at equilibrium. At their lowest mutation rates, this would have been around 0.4. Hence, the population was very much not at equilibrium, as they state, if there was near zero variance. Secondly, that equilibrium should have been reached at the same time in all their simulations, irrespective of the mutation rate. Equilibrium is reached via coalescence, which takes around 4N generations, not by the mutation rate. Hence, whether the mutation rate is tiny or enormous, it takes exactly the same amount of time to reach that equilibrium. These are the basic principles of population genetics that Sanford appears to be unaware of. Apart from the weirdness around Mendel, the specifications of their model are incredibly restrictive. One, whenever a mutation matching the target sequence appears, all subsequent mutations on the string are halted until that mutation either goes to fixation or is lost. Hence, each mutation must sequentially occur in the population. Second, there's no recombination, so not only must they occur sequentially, they must occur on the same background. No other haplotypes can acquire a different mutation that can later recombine onto the same background. These restrictions, coupled with my suspicion of Mendel as a program, prompted me to try to recreate Sanford's model. Like Lynch, I designated a model that I believe is far more biologically realistic. I used the program SLIM, which is the most widely used forward time simulator. It's freely available and is open source, so you can go and download it right now. Furthermore, since SLIM is so superior to Mendel, I could simulate the actual mutation rates without needing to use arbitrary correction factors. My model works like this. I simulated a population of 10,000 individuals that randomly mate, just like in Sanford's model. Every individual starts with a string of 10 nucleotides, all A's, and the target is a string of all T's. Mutations to either of the other nucleotides, C's and G's, are neutral. Unlike Sanford, I do not prevent back mutation, so a beneficial variant can still back mutate to its worst version. Each time a T is reached, the carrier gains a 5% fitness boost in the heterozygous state and 10% in the homozygous state. I set recombination at a low rate of 1e to the negative 8, which can allow mutations to very rarely move on to the same background. Unlike Sanford, I never pause mutation, so any individual at any time can get the target site. I use 3 different mutation rates, 1e to the negative 7, 1e to the negative 8, and 1e to the negative 9. The average human germline mutation rate is around 1e to the negative 8. Next, I restrict the model for only running for 20,000 generations. Assuming 25-year generation time for humans, that's 500,000 years of evolution. That is, I restrict the model to running for only 8% of the amount of time since we've split from chimpanzees. I want to stress that even this model is silly and unrealistic, okay? Again, evolution is not trying to get any sequence. I was just super skeptical that not only was Sanford's model overly restrictive, but that there's like a bug or some serious flaw in how Mendel is coded that is giving them such ridiculous numbers. So what did I find? I found that for the highest mutation rate of 1e to the negative 7, all 10 replicates of my simulation found the target sequence in 500,000 years. In fact, it generally had fixed 8 of the 10 sites in less than 5,000 generations, or about 125,000 years. For the most realistic mutation rate, it fixed an average of 7.5 out of the 10 sites in 500,000 years. In each of these cases, the rate of fixation was the highest when there were still many sites left to fix, but then eventually a kind of substitution saturation occurred, in which the probability of each subsequent mutation decreased as a function of the number already fixed. Only the lowest rate of 1e to the negative 9 failed to fix the majority of sites on average, fixing only around 2.5 in 500,000 years. By simply doubling the length of the simulation to 1,000,000 years instead of just 500,000, we see that for the realistic human mutation rate, all 10 sites are expected to be fixed orders of magnitude faster than in Sanford's model. Are the basic model differences alone sufficient to explain these dramatically different results? Again, I don't think so. While I relax some of Sanford's restrictive assumptions, it still should not have taken 3 orders of magnitude worth of a difference. The most likely culprit? I think it's the default setting in Mendel of heritability, which decreased the benefit of the new mutations so much that they behaved as effectively neutral. And so what they were really waiting on was not the mutation and selection mechanism, they were just waiting on genetic drift to fix a mutation by chance. Since Sanford and Friends' waiting time paper, there has been one additional paper to sneak its way past peer review. This one occurred in the Journal of Theoretical Biology titled On the Waiting Time Until Coordinated Mutations Get Fixed in Regulatory Sequences by Ola Hofstra, Gunter Beckley, and Anne Gager, all of the Discovery Institute. Their model is only a slightly more complex version of Behe and Snoke's in that it permits overlapping generations in back mutations, but otherwise is hindered by the same sort of assumptions. That is, that evolution is trying to find a certain sequence. They write, quote, "In order to evaluate the chances for a neo-Darwinian process to bring about such major phenotypic changes, it is important to give a rough but reasonable estimates of the time it would take for a population to evolve so that the required multiple genetic changes occur." As far back as the 1930s, Sewell Wright and JBS Haldane showed that mutations are not a seriously rate-limiting step to evolution. In fact, they demonstrated that almost all mutations had occurred several times over in a few generations for populations of more than around 100,000 individuals. Asking about the probability of a specific mutation is misleading. Selection is acting on the suites of changes that occur every generation, not waiting around for a particular mutation to occur. The article in Theoretical Biology now has a disclaimer attached to it after several biologists wrote to the journal expressing their concerns. The editor's note, quote, "Since the publication of the paper, it has now become evident that the authors are connected to a creationist group. Moreover, the keywords intelligent design were added by the authors after the review process during the proofing stage, and we were unaware of this action by the authors." Very sneaky of them. They go on, quote, "We have removed these from the online version. We believe intelligent design is not in any way a suitable topic for the Journal of Theoretical Biology." And that completes the saga of the waiting time problem. As this year marks the 20th anniversary of the Behe and Snoke paper, it seemed fitting that we review this bogus creationist idea, despite being repeatedly debunked, including in the court of law by Behe himself. It still hangs on like a toe fungus you just can't quite get rid of. Thanks so much for being here. I hope you enjoyed this. If you have any questions about the waiting time problem or about how the simulations or the math works out, feel free to drop them in the comments below. I'll include a link to my GitHub in the description of this video, in which I actually have the full simulation set up there that you can find if you wanted to download Slim and run the simulation yourself, as well as all the different results for making the plots that I showed in this video. Again, thanks so much for being here, and I'll catch you next time.